How do mutations in gene networks spark cancer? I'm Isol Shinawi from FAU High School, and my research proposal is titled Simulating the Impact of Gene Regulatory Network Mutations on Cancerous Cell Behavior. Cancer doesn't just appear, but it grows silently from genetic disruptions. So what if we could simulate those changes and predict cancer before it even forms? My research objective is how do specific mutations in GRNs, or gene regulatory networks, influence cancer's behavior in cells. So this diagram shows how a gene regulatory network works. So each gene produces mRNA, which makes a protein, and those proteins go back to regulate other genes. For example, protein 1 activates gene 2, which produces protein 2, and that can influence gene 3. These feedback loops are what make up our GRN. My project focuses on what happens when one of these genes, like gene 1, mutates, disrupting the network and potentially triggering cancer's behavior. Why this research matters. Cancer often begins with tiny, invisible disruptions in our genes, long before symptoms appear. But studying these early mutations in patients is almost impossible, and even lab-based models are expensive, time-consuming, and ethically limited. That's where our simulation comes in. By creating a virtual cell and modeling how mutations affect gene networks, we can safely and efficiently explore the roots of cancer before it takes hold. My methodology is divided into 3 main stages. First, I'll build the gene regulatory network by collecting open source interaction data from platforms like BioGrid. I'll focus on well-known cancer-related genes like TP53 and MYC and map how they influence each other. Then I'll simulate mutations in this network using Python. I'll introduce changes at specific genes and track how the disruption spreads through the system like a domino effect. Finally, I'll analyze the simulated cell behavior and compare it to real-world cancer phenotypes from clinical datasets. This will help validate how well my model actually reflects biological outcomes. Why this project is feasible. One of the most powerful things about this project is how achievable it is. I'm using publicly available datasets and free open source tools like Python to build and test the model. The work doesn't require a wet lab, expensive equipment, or advanced computing resources. It's technically realistic, cost-effective, and completely accessible to a student researcher working independently. What makes this project unique? Many student research projects focus on detecting disease after it's already present, through imaging, AI, or diagnostic tools. But my project is different. It takes a step back and asks a deeper question. How does cancer actually begin? By simulating gene regulatory networks and observing how mutations affect them, I'm modeling disease at the systems level, getting closer to cause, not just correlation. Where can this go next? If successful, this framework could simulate drug response, integrate epigenetics, or even help teach biology. It's modular and ready to evolve far beyond this first cancer model. Thank you for watching and for the opportunity to share my proposal. I believe that with the right question, accessible tools, and a drive to explore, we can simulate the invisible and rethink how we study and prevent disease. I'm excited to continue building that future, one line of code at a time. Thanks again.